Welcome back. This is our fourth segment on gene expression. We've talked about the basics of transcription, and we've talked about translation from a very kind of high perspective, a very 20,000 foot overview, if you'd like to think about it like that. So before we get into the mechanics of translation, it's important for us to talk about how the process of translation takes the information in a gene, or actually the process of translation actually takes the information in an mRNA, I should say, and uses that information to build a polypeptide. And so that relies on us understanding the relationship between DNA and mRNA, and between mRNA and polypeptide, which then becomes protein. So let's take a look at this diagram here and start to work on those relationships. So at the top here, we've got a piece of DNA, and the information in that DNA, in that gene, is going to be read by the process of transcription to produce an mRNA. And so the DNA in the gene has got two strands, the coding strand and the template strand. And it's the information in the template strand which is going to be read by transcription by RNA polymerase to produce the mRNA molecule down here. And so translation, as we talked about just now, will start at this start codon here, and so the ribosome will come along the mRNA in this direction from the 5' end, from the ribosome binding site, and it will hit the start codon. And when it hits the start codon, that says to the ribosome, this is where you start reading information. And you read the first start codon, or the first codon, in a sequence of 3 nucleotides. So that 3 there is called, so this 3 here, I'm going to underline it, that is called a codon or a triplet. And that establishes for the ribosome, here are the next 3 that you read, and here are then the next 3 that you read. So this prevents the ribosome picking something random and then reading from there, because AUG, read followed by AGC, followed by GAC, followed by CCU, will give you the sequence of amino acids, methionine, serine, aspartic acid, proline, and then so on as we go through the mRNA. Now, if the ribosome didn't know that this was the first codon, if it just started anywhere, let's just say it picked this G to start at for some reason. If it picked that G and then read in 3s, it would read GCG, and then the next 3 ACC, and then CCU, and that would give us a different sequence of amino acids, and that would be bad. That wouldn't give us the protein specified by the gene. So the start codon is very important in establishing what is called the reading frame. That is, you read these 3 nucleotides, and then the next 3, and then the next 3, and those are called codons. So if we look at the relationship between the DNA and the RNA, the template strand is at the bottom. So this A here in the mRNA corresponds to this T here. And then this U here corresponds to this A here. And then the G corresponds to the C up here. So you can see the relationship between the template strand and the mRNA. So the mRNA is anti-parallel and complementary to the template strand in the gene. And so then we read the information in the mRNA during translation to specify the codon, to specify the amino acids methionine, serine, aspartic acid, and proline. And the way that the ribosome decodes this information in the mRNA is using something called the genetic code. So the genetic code is often misunderstood. People often say to you, what is the genetic code? And people respond by saying it's DNA. DNA is the genetic material. The genetic code is like Morse code. It's used to decipher cryptic information. And so the cryptic information here is the sequence of nucleotides held within the mRNA sequence, and that is complementary to the sequence of the template strand in the gene. And so we're really using the genetic code to help the cell understand the cryptic information in the genes, which is in the language of nucleotides, and so that can be converted into what we know as protein with a sequence of amino acids. So it's a way, literally, of translating the information, the cryptic information in the DNA, to functional information or functional material in proteins. So let's take a look at the genetic code. Here is the genetic code. This is what the cell is actually using. Obviously, it's not a copy of this, but this is what the cell is using biochemically to understand the information in an mRNA and therefore in a gene. So let's just start with a codon. Let's use this genetic code to actually decode a codon. So let's see if I can write a codon well enough that we can actually read it off the screen. So let's go with an A, which is going to be the first nucleotide. Then let's go with a U, that'll be the second nucleotide, and let's see if I can make a G. So there is a G. So let's say that that is our start codon. That's going to be methionine. So that's our start codon. So when the ribosome hits this, how does it decipher that this means methionine? So think about it like this, that there's a table, a genetic code, a table, it's like a lookup table. And so A is in the first position. And so we look there, we look along this row here, and we say, okay, those are all of the codons that start with an A. And then we've got a U in the second position. And so we find U along the second position, so here it is over here. And so we say, okay, now we're looking for all of the codons that are in this column and which correspond to the row of the first position. So now we're just dealing with this group here. So then G is all the way kind of put over here in the third position. And so then we find in this row and in this column we find what corresponds to G, and so then we're dealing just with this bottom row here, and so we can see that AUG corresponds to methionine, which is the first amino acid in all polypeptides. So let me see if I can, let's clear that off. Let's just go back and then come forward just to clear that off. There we go. Now let's do another one just so everyone's confident. Let's pick A, and let's do another A because those are easy. And let's put C on the end. So let's go with AAC. So we're going to find A in the first position. And so we're looking at this row here. So that's going to be every single codon that starts with A. Then we're going to look for the second position. And now we're looking in this column here. I guess that should also include this one at the bottom. I didn't go far enough down there. So that includes everything down to the bottom of the table. And then in the third position, we're looking for the C. And so we're going across here. And so where all of those boxes overlap, we see that AAC corresponds to the amino acid asparagine. So that's how you decode the information in the mRNA. Now, there's some important things about the genetic code. There are two important things about the genetic code. The first thing is that we say the genetic code is redundant. When we say something's redundant, that means there are multiple ways to do the same thing. So let's just take a look at phenylalanine up here to explain redundancy. So when we say the genetic code is redundant, what that means is that there are two codons that specify phenylalanine. So if a ribosome is reading through a piece of mRNA, and it reads UUU, which is in the top, which is this one here. If it reads UUU, it knows that that specifies the amino acid phenylalanine, and it adds that to the polypeptide that's growing. But it could also encounter UUC and do the same thing. So when we say that the genetic code is redundant, we mean that you can specify the same amino acid, now that's important, the same amino acid with different codons. People often confuse redundancy with ambiguity. The genetic code is never ambiguous. So what that means is, let's take CUC for example. That specifies the amino acid leucine. So if you ever see CUC in a piece of RNA, in a piece of mRNA, that will always specify leucine. So don't confuse redundancy with ambiguity, because the genetic code is completely unambiguous. So it's redundant, but unambiguous. So those are two important things about the genetic code. So the ribosome is basically doing this on the fly, if you like. It's able to decode this information, and it does it using a molecule called tRNAs. And we'll talk about tRNAs a little bit later, probably in our next segment or the one after that. But let's wrap this up here with a few questions. So I've got 3 questions here, and then we'll wrap it up. So here's the first question. Let's see how you get on with this one. Okay, let's go on to our second question. And here's our third question in this series. Okay, so just before we finish up here, in Canvas there are some problem sheets. Work through that problem set and post on the online discussions if you want some help. And we will wrap it up there, and we'll come back and we'll start talking about the mechanics of translation next time.